High-Throughput Screening, HTS, is a methodological approach that enables scientists to conduct millions of chemical, genetic, or pharmacological tests rapidly and efficiently. It leverages automation, miniaturized assays, and large-scale data analysis to identify active compounds, antibodies, or genes that modulate specific biological pathways. By systematically testing a vast array of biological or chemical entities against defined targets, HTS accelerates the discovery process in fields like drug development and molecular biology, fundamentally transforming how initial screening phases are conducted. For instance, pharmaceutical companies utilize HTS to screen extensive libraries of chemical compounds to find potential new drugs. A company may screen over 1 million compounds to identify those that inhibit a particular enzyme implicated in a disease. Automated robotic systems handle microplates containing thousands of tiny wells, each well representing a unique test condition. This process enables the identification of lead compounds in weeks rather than years, significantly expediting the drug discovery pipeline. Imagine HTS as casting a vast net into the ocean to catch fish. Instead of fishing with a single line, testing one compound at a time, HTS allows scientists to deploy a massive net that captures thousands of fish compounds simultaneously. This approach vastly increases the chances of finding the big catch, the compound with the desired biological activity, much more efficiently than traditional methods. HTS has become indispensable in modern research due to its ability to rapidly identify biologically active compounds from large libraries, thereby accelerating the development of therapeutics and advancing our understanding of biological systems. It facilitates the systematic exploration of chemical space and genetic variations, enabling the discovery of novel drug candidates, biomarkers, and molecular targets. HTS also supports the functional analysis of genes and proteins, contributing to fields like genomics and proteomics by providing high-throughput methods to study complex biological interactions. During an emerging infectious disease outbreak, researchers can employ HTS to quickly evaluate existing drugs for efficacy against the new pathogen. By screening a vast array of compounds, they might identify antiviral agents that can be repurposed, thereby expediting the development of treatments and potentially saving lives. This rapid response capability is critical in addressing public health emergencies. Think of HTS as using a sophisticated metal detector to find valuable coins buried in a large field. Instead of digging randomly and slowly across the entire area, the metal detector quickly scans the field, pinpointing the exact locations where the coins, active compounds, are buried. This targeted approach saves time and resources, leading to faster and more efficient discoveries. Traditional HTS methods, while revolutionary, face significant limitations related to resource consumption, cost, and scalability. These methods often require large volumes of reagents and samples, leading to substantial expenses and waste. The physical size and throughput of multi-well plates are constrained, limiting the number of assays that can be performed simultaneously. Additionally, the handling and dispensing of small volumes at high speeds can result in technical challenges such as evaporation, cross-contamination, and inaccuracies, which may compromise data quality and reproducibility. A biotech company conducting HTS might need to use liters of costly reagents and cell cultures to screen their compound library. The high reagent costs and the need for extensive automation infrastructure can make such screenings prohibitively expensive for smaller laboratories. Furthermore, the large volume of waste generated poses environmental concerns and requires proper disposal, adding to operational complexities. This scenario is similar to watering a small garden using a fire hose. The equipment is not appropriately scaled for the task, resulting in overuse of water, potential damage to the plants, and unnecessary waste. Similarly, traditional HTS methods can be inefficient and resource-intensive when not optimally designed for the scale of modern screening needs. Microfluidics involves the precise control and manipulation of fluids at the microscale, typically in channels with dimensions ranging from tens to hundreds of micrometers. At this scale, fluids exhibit unique behaviours dominated by laminar flow, surface tension, and capillary forces rather than turbulence. Microfluidic systems exploit these properties to enable the handling of extremely small volumes of liquids, nano to picolitres, facilitating highly controlled chemical and biological reactions. The integration of multiple laboratory functions on a single microchip, lab on a chip, allows for complex analyses with minimal sample and reagent consumption. A practical application of microfluidics is in point-of-care diagnostics, where a handheld device can perform blood tests using a single drop of blood. The microfluidic chip within the device orchestrates sample preparation, mixing with reagents, and detection of biomarkers, providing rapid results. This technology is invaluable in remote or resource-limited settings where traditional laboratory infrastructure is unavailable. Microfluidics can be likened to building intricate miniature roadways for fluids, much like the intricate circuits on a microchip guide electrical currents. Just as electronic microchips control the flow of electrons to perform computations, microfluidic chips control the flow of fluids to perform chemical and biological processes with high precision and efficiency. Microfluidic platforms offer several advantages over traditional macroscale systems. They significantly reduce the volume of reagents and samples required, lowering costs and minimising waste. The high surface area to volume ratio enhances reaction kinetics and heat transfer, leading to faster and more efficient reactions. Microfluidics allows for precise control over fluid flow, mixing and environmental conditions, improving the reproducibility and sensitivity of assays. Additionally, the ability to integrate multiple functions on a single chip enables automation and parallel processing, increasing throughput while reducing the potential for human error. In pharmaceutical research, microfluidic devices can perform high throughput screening using only nanoliters of reagents per assay, compared to microliters in traditional systems. This reduction can translate into thousands of dollars in savings per screening campaign, especially when using expensive biological reagents like enzymes or antibodies. The increased efficiency also allows for more assays to be conducted within the same time frame, accelerating the discovery process. This advantage is comparable to using energy-efficient LED bulbs instead of traditional incandescent bulbs. LEDs consume less power, last longer, and provide the same or better quality of light. Similarly, microfluidic systems achieve the desired experimental outcomes more efficiently and sustainably than their macroscale counterparts. Integrating microfluidics with HTS combines the scalability and efficiency of high throughput methods with the precision and resource economy of microfluidic technology. This synergy addresses the limitations of traditional HTS by enabling assays to be conducted at significantly smaller scales without compromising data quality. Microfluidic HTS platforms can perform thousands of simultaneous assays on a single chip, each in isolated and controlled environments. This integration enhances automation, reduces the potential for cross-contamination, and allows for real-time data acquisition and analysis. Researchers might develop a microfluidic HTS platform that uses droplet-based technology to encapsulate individual reactions within nanoliter-sized droplets. Each droplet contains a different test compound and a biological target, such as an enzyme or receptor. The platform can generate and analyse millions of these droplets rapidly, identifying active compounds with high sensitivity and specificity. This approach streamlines the screening process and enables the exploration of larger chemical spaces. Integrating microfluidics with HTS is like upgrading from batch cooking in a large pot to using a high-tech food processor that can handle multiple ingredients separately but simultaneously. The food processor allows for precise control over each component, mixing and processing them efficiently without cross-contamination, resulting in a better quality final product in less time. Several microfluidic technologies are utilised in screening applications, each with unique capabilities. Droplet-based microfluidics generates and manipulates tiny droplets within an immiscible carrier fluid, effectively creating thousands of miniature reaction vessels. Continuous flow microfluidics controls the steady flow of fluids through microchannels, enabling the formation of gradients and precise mixing of reagents. Digital microfluidics manipulates discrete droplets on an array of electrodes through electrowetting, allowing for programmable and reconfigurable assays without the need for physical channels. These technologies expand the versatility and efficiency of screening applications. In drug discovery, droplet-based microfluidics can be used to perform enzyme inhibition assays. Each droplet contains the enzyme, substrate, and a potential inhibitor. By monitoring fluorescence changes within each droplet, researchers can determine which compounds effectively inhibit the enzyme. The high throughput of droplet generation allows for the screening of extensive compound libraries in a fraction of the time required by traditional methods. These microfluidic technologies are like different types of assembly lines in a factory. Droplet-based microfluidics is akin to having individual workers, droplets, assembling products independently, allowing for customization and high throughput. Continuous flow microfluidics resembles a conveyor belt system where products move along a line, receiving inputs at various stages. Digital microfluidics is comparable to robotic arms moving components freely on a grid, providing flexibility and precision in assembly. Microfluidic HTS platforms have broad applications across various scientific disciplines. In drug discovery, they facilitate rapid screening of potential therapeutics with improved accuracy and lower costs. In genomics and proteomics, microfluidics enables high throughput analyses of genes and proteins, such as sequencing, expression profiling, and interaction studies. For cell-based assays, microfluidic devices can simulate physiological conditions more accurately than traditional cultures, allowing for studies of cellular responses, toxicity testing, and the development of organ-on-a-chip models that mimic human tissues. An application in toxicology involves using microfluidic devices to assess the cytotoxic effects of environmental chemicals on human liver cells. By exposing cells cultured in microfluidic chambers to different concentrations of chemicals, researchers can monitor cellular responses in real time. This method provides more physiologically relevant data compared to traditional static cultures and can improve the prediction of human responses to toxins. Utilising microfluidic HTS platforms is like employing a flight simulator for pilot training. The simulator provides a realistic and controlled environment where pilots can experience various scenarios without the risks and costs associated with actual flight. Similarly, microfluidic devices offer a controlled and realistic setting for biological assays, enhancing the relevance and safety of experiments. Recent advancements in microfluidic HTS have led to significant breakthroughs in research and technology. Innovations include the development of high-throughput single-cell analysis platforms, integration with mass spectrometry for direct detection of analytes, and the creation of complex tissue models on chips. Case studies have demonstrated the successful application of microfluidic HTS in identifying novel drug candidates, understanding disease mechanisms at the cellular level, and performing comprehensive genetic screens with unparalleled efficiency. A notable case study involves using microfluidic devices for personalised cancer therapy. Researchers have developed a microfluidic platform that tests the sensitivity of a patient's tumour cells to various chemotherapy drugs. By analysing the cellular responses within the microfluidic device, clinicians can identify the most effective treatment regimen for the individual patient, improving therapeutic outcomes and reducing unnecessary side effects. These advancements are akin to moving from standard definition to high definition television. The increased resolution provides more detail and clarity, enhancing the viewing experience. In microfluidic HTS, high resolution at the cellular or molecular level provides deeper insights into biological processes, leading to more informed decision-making in research and clinical applications. Microfluidic HTS offers numerous advantages, including reduced reagent consumption, lower operational costs, increased throughput, and improved assay sensitivity and specificity. The technology enables high-density screening with minimal cross-contamination and allows for complex assays that mimic physiological conditions more closely than traditional methods. However, challenges exist, such as the complexity of device fabrication, potential issues with clogging or bubble formation in microchannels, and the need for specialised equipment and training. Data management also poses a challenge due to the large volumes of data generated, requiring robust analytical tools and computational resources. While a microfluidic HTS platform can dramatically increase screening efficiency, a laboratory may encounter difficulties in integrating the technology into existing workflows. The need for specialised fabrication facilities and expertise in microfluidics can be barriers to adoption. Additionally, ensuring consistent device performance and reliability requires rigorous quality control and validation processes. This situation is similar to adopting a new, advanced software system in a company. The software promises increased productivity and efficiency, but it requires staff training, updates to infrastructure, and troubleshooting of initial technical issues. The long-term benefits are substantial, but the transition period involves overcoming practical challenges. To address these challenges, researchers and engineers are developing standardized fabrication processes and modular microfluidic platforms that simplify device production and operation. Advances in materials science, such as the use of biocompatible and anti-fouling coatings, reduce issues with sample adhesion and channel blockage. The implementation of automated control systems and user-friendly interfaces makes microfluidic devices more accessible to non-specialists. Moreover, integrating advanced data analytics and machine learning algorithms helps manage and interpret the complex datasets generated by microfluidic HTS, enhancing the extraction of meaningful insights. A company might introduce a microfluidic HTS system with pre-fabricated, interchangeable chips and plug-and-play functionality. This system reduces the need for custom device fabrication and allows researchers to focus on experimental design and data analysis. The inclusion of software that automates data processing and provides intuitive visualization tools further streamlines the workflow. Implementing these solutions is like transitioning from manual car assembly to an automated production line. Automation increases consistency, efficiency, and scalability while reducing the potential for human error. In microfluidic HTS, automation and standardization simplify operations and make the technology more accessible to a broader range of users. Looking ahead, the integration of microfluidic HTS with emerging technologies such as artificial intelligence and machine learning is expected to revolutionize the field. AI can optimize experimental design, predict outcomes, and automate data interpretation, leading to smarter and more efficient screening processes. The continued development of organ-on-a-chip models and personalised medicine applications will enable tailored therapeutic approaches based on individual patient data. Efforts to standardise microfluidic technologies and establish regulatory frameworks will facilitate their adoption in clinical and commercial settings, ultimately improving healthcare outcomes. In the future, a clinician might use a microfluidic device integrated with AI to test a patient's response to various medications in real time. The system could analyse the data and recommend the optimal treatment based on predictive modelling and comparison with large datasets of patient outcomes. This personalised approach enhances treatment efficacy and reduces the trial and error aspect of prescribing medications. This progression is similar to the evolution of navigation systems in vehicles. Early maps provided basic guidance, while modern GPS systems offer real-time traffic updates, route optimisation, and predictive arrival times. The integration of advanced technologies enhances functionality and user experience, just as AI and machine learning will enhance microfluidic HTS capabilities. In conclusion, microfluidic platforms represent a transformative advancement in high-throughput screening, offering significant benefits in terms of efficiency, cost-effectiveness, and data quality. By addressing the limitations of traditional HTS methods, microfluidics enables more precise and scalable assays, fostering innovation in drug discovery, diagnostics, and fundamental research. While challenges remain, ongoing technological developments and interdisciplinary collaboration are paving the way for widespread adoption. The future of microfluidic HTS is promising, with the potential to significantly impact healthcare and scientific discovery. As microfluidic HTS technologies become more robust and user-friendly, they are likely to become standard tools in laboratories worldwide. This accessibility will democratise high-throughput experimentation, allowing researchers from various disciplines and resource levels to contribute to scientific advancements and address global health challenges more effectively. Embracing microfluidic HTS is akin to adopting renewable energy sources. While initial investments and infrastructure changes are necessary, the long-term benefits of sustainability, efficiency, and positive impact are substantial. Similarly, microfluidic HTS promises to create a more efficient and innovative landscape in scientific research and development.